Your brain is physically shrinking right now if you are grieving. Not metaphorically, not emotionally, structurally. At this very moment, if you have lost someone you love, the gray matter in your prefrontal cortex is measurably thinner than it was before the loss. Scientists at Harvard Medical School have confirmed this, and what is happening inside those cells, at the molecular level, is one of the most astonishing and least understood processes in all of human biology. Most people will never know this. Most doctors will never tell you this. But you deserve to understand exactly what is happening inside your own brain. We are not going to talk about the 5 stages of grief today. You've heard those. What we are going to do is go somewhere most people never go. We are going to zoom all the way down, past the emotions, past the feelings, past the thoughts themselves, and look at what is actually happening inside your neurons, your glial cells, your synaptic connections, when the person you love is no longer there. Because what grief does to your brain is not a metaphor, it is a physical event, a measurable biological cascade that begins in the first seconds after loss and continues reshaping your neural architecture for months, sometimes years afterward. And understanding it, truly understanding it, at the level of molecules and mechanisms, changes everything about how you see yourself in the middle of it. Before we go further, a brief but important note. Everything you are about to hear is drawn from peer-reviewed research published in journals including Nature, the Journal of Neuroscience, JAMA, and findings from Harvard Medical School, the NHS, and University College London. This video is for educational purposes only and is not a substitute for medical or psychological advice. If you are in the depths of grief right now, please speak with a qualified professional who can support you properly. What science can offer is understanding, and sometimes understanding is the very beginning of healing. Now, let us go inside. The moment you receive devastating news, the moment you learn that someone you love is gone, your brain does something extraordinary. Within milliseconds, your amygdala, the brain's ancient alarm system buried deep in the temporal lobe, fires an emergency signal of extraordinary intensity. This is not a slow, considered, conscious response. This is primal, electrical, and instant. The amygdala screams to the hypothalamus, a small but extraordinarily powerful structure at the base of the brain, which immediately triggers the release of corticotropin-releasing hormone, CRH. CRH floods into the pituitary gland, which then sends its own hormonal signal cascading down to the adrenal glands sitting atop your kidneys, commanding them to flood your bloodstream with cortisol and adrenaline. This entire sequence, from the moment of devastating news to the first surge of stress hormones entering your blood, takes less than 2 seconds. Scientists call this the HPA axis response, the hypothalamic pituitary adrenal cascade. And in grief, unlike in ordinary stress, this cascade does not switch off after a few minutes. It runs, and it runs, and it keeps running for weeks, months, sometimes years. Here is where it becomes remarkable at the cellular level. Cortisol, in small doses and short bursts, is one of the most protective molecules in your body. It sharpens your focus, mobilizes glucose from your liver, suppresses non-essential biological functions, and prepares every system in your body to respond to immediate threat. In the right amounts, at the right moments, cortisol is life-saving. But cortisol in sustained, unrelenting, grief-driven levels becomes something else entirely. It becomes neurotoxic. Research published in Nature Neuroscience has demonstrated that chronic cortisol exposure causes dendritic retraction. Your neurons, the roughly 86 billion nerve cells that form the physical substrate of everything you think, feel, remember, and are, have long branching arms called dendrites. These dendrites are the receiving antennae of your brain cells, the elaborate structures through which neurons communicate with one another across the tiny gaps we call synapses. Under sustained cortisol exposure, those dendrites physically shrink. They pull back. They retract like a frightened animal withdrawing from danger. And when the dendrites retract, the synaptic connections between neurons, the bridges across which memory, emotion, language, and thought travel, begin to thin, weaken, and in some cases, collapse entirely. This is not a minor or symbolic process. Studies indicate that in severe or prolonged grief, the hippocampus, the seahorse-shaped structure nestled deep inside your temporal lobe, responsible for memory consolidation, spatial navigation, and emotional regulation, can lose measurable volume. Research from University College London using high-resolution MRI imaging has shown hippocampal shrinkage in individuals experiencing complicated grief. The prefrontal cortex, the brain region that governs rational thought, long-term planning, impulse control, and the capacity to regulate emotion, thins under sustained cortisol exposure. This is the precise neurological reason why grieving people cannot think clearly, cannot make simple decisions, cannot remember where they put their keys or what day it is, cannot stop themselves from crying at unexpected moments. It is not weakness. It is not a failure of character or resilience. It is cortisol physically remodeling their brain architecture at the cellular level in real time. But here is what almost no one knows about what happens next. And this is where the science becomes truly extraordinary. Grief does not only damage neurons through cortisol, it triggers a second, parallel process called neuroinflammation. And understanding this changes the picture completely. When the brain is under sustained psychological stress, the immune cells of the brain, specialized cells called microglia, shift from their resting surveillance state into an activated inflammatory state. Microglia make up roughly 10 to 15% of all cells in the brain. In their resting state, they are the brain's custodial crew, constantly patrolling the neural landscape with extraordinary precision, clearing cellular debris, pruning old and inefficient synapses, wrapping protective myelin around nerve fibers, and supporting the overall health and architecture of the neural environment. They are, in every meaningful sense, the gardeners of your brain. But when cortisol floods the system month after month, as it does in grief, microglia become reactive. They abandon their gardening role and shift into an inflammatory, defensive posture. They begin releasing inflammatory proteins called cytokines, specifically interleukin-6, interleukin-1 beta, and tumor necrosis factor alpha. These cytokines, research published in JAMA and the Journal of Neuroscience has confirmed, directly suppress the synthesis and release of serotonin and dopamine. They interfere with the reuptake mechanisms that regulate mood. They alter the sensitivity of receptors throughout the limbic system. This is why grief so often feels clinically indistinguishable from depression, because at the molecular level, grief is activating many of the same neuroinflammatory and neurochemical pathways. The sadness of grief is not only psychological, it is cytokine-driven. It is a measurable immune response unfolding inside your skull. Let me give you a Feynman analogy to make the invisible visible. Imagine your brain as a vast, luminous city of 86 billion lights, each neuron a lamp, each synapse a wire connecting them, each thought a pattern of light moving across the city in real time. In a life filled with connection, love, and meaning, this city blazes. Every district is lit. The streets hum. When grief strikes, it is as though the city's power grid is suddenly, catastrophically flooded. Lamps flicker, wires corrode, whole districts dim. And the maintenance crew, the microglia, who should be out there in the streets repairing those wires and trimming overgrown connections, have instead been mobilized as emergency inflammatory responders. They are throwing alarm signals in every direction, trying to protect a city that is not under physical attack. Because your ancient, evolutionarily conserved brain cannot distinguish between the loss of a beloved person and the approach of a physical predator. The grief response is a survival mechanism, misfiring in a world where the threat cannot be fought or fled. Now, in this dimension of the science is critical. After the age of 55, this entire process changes in ways that most people are never told. Research suggests that the aging brain already produces higher baseline levels of inflammatory cytokines, even in the absence of stress. The microglial cells in the brain over 55 are already somewhat primed toward the activated inflammatory state simply as a function of normal biological aging, a process scientists call inflammaging. This means that when grief strikes someone in their 60s or 70s, the neuroinflammatory response is typically more pronounced, more rapidly established, and significantly harder for the brain's own regulatory systems to contain and resolve. The AHA and NHS have both published data indicating that prolonged grief in older adults is associated with a dramatically elevated risk of cardiovascular events, heart attacks, and strokes in the 12 months following bereavement. And this is not a coincidence or a statistical curiosity. It is a direct mechanism. The neuroinflammation triggered inside the grieving brain does not stay in the brain. Those cytokines enter systemic circulation. They inflame the endothelial lining of blood vessels throughout the body. They alter coagulation pathways, promoting clotting. They destabilize arterial plaque that has accumulated over decades. They elevate blood pressure through sustained activation of the sympathetic nervous system. The broken heart is not a metaphor. It is a vascular and immunological event with a precise neurological origin that begins in the amygdala and ends in the coronary arteries. And yet, and this is where the science delivers something genuinely, profoundly extraordinary, the brain retains the capacity to rebuild even after all of this. The same hippocampus that shrinks under grief can regenerate. The process is called adult neurogenesis, the birth of entirely new neurons in the adult brain. And while scientists once believed this process ceased in early adulthood, research now published in Nature Medicine confirms that new neurons continue to be born in the hippocampal dentate gyrus well into old age. The key molecular trigger for this neurogenesis is a protein called BDNF, brain-derived neurotrophic factor. BDNF is, in the most literal sense, the brain's own fertilizer. It promotes the survival of existing neurons, stimulates the growth of new synaptic connections, and directly supports the birth of new hippocampal cells. And here is the embarrassingly simple, almost insultingly straightforward truth that the research consistently points toward. Physical movement, even a brisk 30-minute walk taken 3 times a week, produces measurable, statistically significant increases in circulating BDNF. Research from the University of British Columbia has confirmed that regular aerobic exercise increases hippocampal volume even in adults over the age of 60. Not a cure, not a reversal of all that cortisol has done, but a genuine, verifiable, molecular act of repair occurring inside your grieving brain every single time you move your body through space. Zoom out now. All the way out. Consider for a moment that the atoms currently composing your neurons, the carbon, the hydrogen, the oxygen, the nitrogen, were forged in the nuclear cores of dying stars billions of years before the Earth existed. The same atoms that built the person you are grieving. The same atoms that are building you right now, in this moment. The universe does not grieve the way we do, because at the fundamental level, the universe does not lose anything. It only transforms. What feels like devastating, permanent absence is, at the atomic level, a redistribution of matter that has been cycling through stars and oceans and living things for 13 billion years. And somewhere in that understanding, not as a comfort that erases grief, but as a truth that sits alongside it, there is something worth returning to. If this video reached something in you today, if it gave language or structure to something you have been living through without words, please send it to someone who needs to hear it. Share it on WhatsApp, share it on Facebook, forward it to a friend who is quietly carrying loss and does not know why their thinking feels so slow, why their memory feels so unreliable, why their body feels so exhausted. Sometimes the most healing thing we can offer another human being is this simple message. What you are feeling has a biological name, and science has looked inside it, and it is real, and you are not broken. The question I want to leave you with today is this. If grief physically remodels the brain, if it literally reshapes the architecture of thought, memory, and emotion at the cellular level, then what is the brain that comes through it on the other side? What does recovery actually build? What does the neuroscience of resilience look like at the molecular level? Next time, we go inside that question. And what we find there is not what most people expect.